are going to go through a little bit of lecture today, dialysis and osmotic pressure. You should have been finishing quiz 7.6. You make sure that you're redoing your quizzes. Do not do any quizzes, because I'm going to go to the point where if you don't take the quiz in class, at least try it, then I'm not going to let you retake it. It'll just be a 0. Okay, so make sure you at least try it, and then you can retake it as many times as you want up to the day of the test, okay? It's just getting to be a pain in the butt, because I have to go back and look at those every single time and try and, there's all of you see 11, 10 kids in one big Excel spreadsheet that I have to try and figure out your grades for each quiz, and it's a lot easier if it's, I just, you know, I'm able to put stuff in right at first. So make sure you're checking that. We are going to finish our lab today on colligative properties. We'll talk about that. The test opens today and is open through Tuesday next week. So make sure that you are understanding, making sure you've got your sapling done, taking the pretest like a pretest, like take it like a test, and then figure out what you didn't understand and what you need to go back and study and what you need help with before you go take the test. I can't stress that enough. You're going to have some time to work and finish your lab notebooks today. If you finish and get them done, turn them in. They are due next time. Next time is a work day. So Friday when you come, it is a work day. Clean it up, get questions asked, do whatever you need to get done to be taking that test and move on. Tuesday we'll be moving on with a new chapter. Okay? So that's what's going on there. Let's go ahead and go to our lecture. So, like I said, we're going into dialysis and osmotic pressure. Lab notebooks are due next time. You should be finishing sapling if you haven't already. We're going to be doing the application part of colligative properties today, which is making ice cream, and I'll show you how to go through that. It is a fun thing that you can do on dates and all kinds of stuff, and I'll show you how to adjust it so you can do it. So, going forward, I've got, come on, I'm at the very end. I forgot to reset this, so just, okay, this is a lot of definitions on this one. So as we go through the definitions on this, fill in your lab or your study guide as best you can. If you have questions, you don't have to write every word down, but you need to make sure you get the gist of it. So we talked about colloids. Tell me what colloids are in your own words. What are they? Solutions that scatter light. So it's really not a solution, because a true solution, the particles are really small, right? So because they're really small, they don't scatter light. So it is a suspension or a mixture where the particles stay in solution, but that when I put a light through it, I get scattered light, which is known as what? Tyndall effect. Okay. So in a colloid, instead of having a solute and a solvent, we have a dispersing media and a dispersed phase. Same kind of concept here, but a solute and a solvent only are for a true solution. So the dispersing media is like a solvent. It's the thing that disperses whatever your solute, which is the dispersed phase is. Okay? They just use different verbs or different vocabulary for it. Yes? It's like the solvent for a colloid. Okay? So we don't call it a solvent when it's a colloid. We call it a dispersing medium. And we don't call it a solute for a colloid. We call it a dispersed phase. Okay? Because it's a bigger particle. Okay? So that's basically what that means. So when you're talking about a colloid, you're talking about a two-phase system. One that's a dispersing medium and one that's a dispersed phase. Okay, still the same sort of as solvent or a solution where you have a solute and solvent, but the particles are bigger. Okay, everybody okay with that? All right, moving forward. So remember your Tyndall effect where you can see the light and it sort of lights up the whole thing. When it's not a Tyndall effect, it's just the bending of the glass on the end, the light. Okay, then we go to a couple new words or things that we need to define. An emulsifying agent is what is often used in soap. It's what pulls together oils and non-oils. Okay, so if I have an emulsifying agent and I want two things that wouldn't normally mix, like oil and water, to mix, I put in an emulsifying agent so they will stay mixed together. okay? Because it has a polar end and a nonpolar end, or a water end and a fat end, it wants to be dispersed universally. So we call it an emulsion when we do that, and we use an emulsifying agent. And then stabilizing agent just is what it says. When you stabilize something, what do you do? You stabilize it. What does stabilize mean? Yeah, keep it under control, or keep it contained. So a stabilizing agent stops a reaction from happening between two chemicals. It can also be thought of as the opposite of a catalyst. Okay, so a stabilizing agent stops the reaction from happening. So, for instance, when your mom doesn't want you to date before 16, so she might put a stabilizing effect in there, like you can't go to these people's houses if there's mixed people there. That would be a stabilizing effect. Okay, so this is sort of opposite of a catalyst, and this helps us to also inhibit separation of suspensions, emulsions, and foams, or colloids. We usually use it more with suspensions. They use them a lot with like Cool Whip. We don't want it to fall apart. We want it to stay as Cool Whip and pretty and nice. We don't want it to go to the oils and the other stuff that was in there. And if you have old Cool Whip and you open it up, it has destabilized and gone to the oils and nasty. Okay, so then we get to a suspension, and we've worked with suspensions. What are suspensions? It settles out. The particles are big. Do we get a Tyndall effect with suspensions? Yes, we do. Okay, as long as it's suspended, right? If it's fallen out, then we don't. Yes. So a multiplying agent will take things that are opposite, so a polar and a nonpolar, and make them be able to mix and stay mixed. So we often use emulsifying agents when we're using soaps, because they'll have one end that will attract to salts and one end that will attract to oils. So soaps have emulsifying agents in them. Okay? All right. And if you go on to 1120, we do a lot of organic stuff and biochemical stuff. We don't do a lot of math in 1120. It's all about how things go together and how does things react in your body, and how do oils work, and how does soap work, and how do polymers work, and making polymers, and how do you do if you're going to be a nurse, how do I make an isotonic solution that is not going to send somebody into shock, and how do I do all this? That's what 1120 is about. Okay, it's not moles, it's not molar, it's not that kind of stuff. It's more visual, more putting things together that way. Okay. All right. So going forward, we talk about dialysis. What dialysis is a colligative property of osmosis in use. Okay, where we actually use it. And when you have dialysis, you have somebody attached in one vein, artery or vein, I can't remember which one it is, and then it comes out the other side. So it's circulating the blood through an outside dialysis machine. And the dialysis machine has our dialysis tubing in it. Now, it's a lot fancier dialysis tubing than what we used in the back, but it will allow the waste, the urea waste from the blood to come out of the blood, but the blood cells to stay in. So it cleans the system as it goes through. And as we go back and we look at our lab that we did last time, and we put stuff in salt solution, we put stuff in sugar solution, we put stuff in water, and we're going to see some things grew, some things shrunk. And that's what is going to happen with dialysis. It's actually pushing in some saline solution that's clean and pulling out the dirty. So it's sort of like a wash of the blood. And I've put a lot of that on the right-hand side of your notes. Now, if you don't get a good connection, you end up with stenosis up here, which is hardening of the arteries. And if you try and push a solution through that is not isotonic with the blood in your system, that can cause hardening of the arteries. Okay, so for instance, I'm diabetic, and they put me at the very end of a surgery thing when I had to do surgery, and my sugars got really, really low. and then they had to push sugar through. Well, when they pushed sugar through, they actually fried my vein because they pushed it too fast. It wasn't isotonic with what my blood was, and so it fried my brain. Yeah, it fried my brain. Yeah, you can tell. It fried my vein that they had tried to put my IV in, and they actually had to move my IV for surgery because that vein was gone. So it's really important. Okay, so as we're doing things in 1110, 1120, we're working more towards the medical field. So as you go on, if you go on to 1110 or 1120, we actually do work with, we worked with salt and we worked with sugar here. But as you go on to 1120, we now start working with starch and urea. Urea is a molecule that our body makes to help get rid of the bad stuff. We'll work with albumin, we'll work with enzymes, we work with carbohydrates and proteins and all that fun stuff. Okay? And you're going to go back and see what happens when it's reached equilibrium. And you'll notice, see, this would be the waste that we're trying to get rid of in the bloodstream, and if we put it in something that doesn't have any of the waste, then it will come through. The small particles will come through. So if you've ever been in a bathtub and you sat there too long, what happens? You start to shrivel up because your solution's trying to go out and make the solution of the bathtub the same salinity as what you have inside you. But if you put bath salts in, then it doesn't happen because you're the same inside and out. Okay, so this is really important with the medical because if I put the wrong solution in, I can actually make blood cells explode or implode. So if I put too much salt in something that's going in, I can actually make the blood cells try to dilute the solution that's coming in, and that implodes them. If I make the solution too much water and not enough salt, then I can actually blow up the blood cells because they'll try and go the opposite way. So you have to be really careful, and there's a very, very thin line of what we can use with saline. Okay? So that's the end of today. I'm going to walk you through what's at the back. You're going to go back in the back, and I know some kids, if this period, it was so fun. Your prunes are going to have grown, and they're going to look gross, but it's okay to pull those things out with your hands. You got to dry them off with a paper towel, not it. And then you're going to weigh them. Okay, shh. Once you weigh them in your dialysis tubes and put the numbers down, then go ahead and throw those prunes and the dialysis tubes away. Clean up your beakers. The beakers are going to be put back on the shelf. So I'm going to have you clean them up, put them back on the shelf, and then you're going to get a stamp from me. When you do that, you're going to come back in. So having